that introduction. So my name is Carey. I am a member of the Rosenheim Lab, and today I'll be talking about the effectiveness of mangroves as carbon sinks. And really I wanted to add a secondary title asking, are we really using the correct methods to answer this question? So to start off with, where's our current knowledge on this topic? And so for this figure, on our x-axis, we have various ecosystems, and our y-axis, we have average global carbon stocks. So this is how much each of these ecosystems is storing of carbon. And a lot of this has to do with something called blue carbon, which I'm sure some of you are familiar with. And blue carbon ecosystems are just coastal ecosystems that store a lot of carbon. So that can be anything from a tidal marsh to a seagrass bed to a mangrove. And we can see that mangroves store substantially more carbon, both above ground and below ground, than not only other blue carbon ecosystems, but other ecosystems that are terrestrial, such as evergreen forests. In addition to that, mangroves are expected to expand in the future. So this is an aerial image from the Merritt Island Wildlife Refuge on the east coast of Florida. You can see our mangroves here in orange, and our salt marshes are shown in green. And so from 2003 to 2010, there's a clear expansion from mangroves, and this is due to the warming of our planet. So as the planet warms and sea level rises, the areas in which mangroves can live is actually going to expand. And so that means we can store more carbon, which is good news for our planet. And this is where the question comes in. So we know mangroves are storing a large amount of carbon. However, less work has been done about on what time scale is this carbon being stored. And really, this is the key to understanding the effectiveness of this sink. And in order to answer this question, we need to use a few different methods. And so I wanted to use two different chronometers or measures of time. And so the first chronometer I'm using is lead-210 dating. And so lead-210 gives us the ability to get precise sediment dates. And so when looking at these figures, you can always think about our lead-210 dates being on our x-axis. And the second chronometer I wanted to use was bomb radiocarbon. And so I think of bomb radiocarbon as being kind of a separate subset of radiocarbon from what you traditionally think of. So bomb radiocarbon was produced anthropogenically. It peaked in about the late 1960s due to nuclear weapons testing. So we don't use it in the way that we traditionally would for dating things. Because of this big artificial spike, it would kind of mess up our dates. So instead we can use that as a tracer. So a lot of people use it as an oceanic tracer. In this case, I'm using it as a tracer of soil organic matter cycling. So you can always think of it on our y-axis over here. So let's say we go out to our mangrove, we collect a sediment core, and we want to understand what would our radiocarbon signal look like in our sediment core if our soil and atmosphere were in perfect equilibrium. And it would actually track exactly along with this atmospheric bomb curve because we have the fluid exchange of carbon in between our atmosphere and our soil. However, we know that this isn't always the case. So like the ocean, the soil tends to be out of equilibrium with the atmosphere. And in order to understand this, we need to look at turnover time. And turnover time simply refers to how long is the carbon staying in the soil itself. So we know that carbon enters the soil in the form of leaves, detritus or tidal DOC. While it's in the soil, it's going to be processed by microbes, and it will be respired as carbon dioxide. So carbon that enters and leaves quickly is going to have a rapid turnover time, and carbon that enters and is stored for a while is going to have a slow turnover time. So in a more realistic situation where we have our soil and our atmosphere out of equilibrium, what would it look like if we had the rapid exchange of carbon between our soil and atmosphere, or a rapid turnover time? In that case, our radiocarbon would look something like this. So we still see kind of that distinct atmospheric bomb curve, however, it's not tracking exactly, and that's due to that disequilibrium between our soil and the atmosphere. And in this case, we consider it to be a bad carbon sink, and that's simply because our carbon is coming in and it's leaving pretty quickly, so it's not being stored for a significant amount of time. In a third situation, we might have our soil and atmosphere still out of equilibrium with one another, but we'd have this exchange of carbon between our soil and the atmosphere, so we'd have a slow turnover time. In this case, the radiocarbon signature in our core would look something like this. So we see a much more elongated shape. We don't really see that distinct bomb radiocarbon peak, and that's due to, again, that disequilibrium and the time that it takes to incorporate carbon into that sediment column. But we consider this to be a good carbon sink because the carbon is being incorporated and then it's staying there for a long period of time. So to get into some of our data, we collected 2 sediment cores from 10,000 islands in southern Florida, and we did both lead-210 dating and radiocarbon dating. And for our initial data, one of the first things that we noticed were these 3 points here. And they were a little bit odd because they corresponded to lead-210 dates that were before 1950. However, they lied above this line on our bomb radiocarbon axis. And everything that's above this 0 line on our radiocarbon axis is radiocarbon that was produced after 1950. So we had lead-210 that was pre-1950 and radiocarbon that was post-1950. So at first we thought we might have contamination in our samples, and after testing for contamination and realizing we didn't, we actually realized that we had some young carbon at depth. So what was happening was that we had young carbon at the top of our core, it was being transported down to the bottom of our core, and that was making our radiocarbon ages appear younger than they were. The second thing we noticed was that these curves actually looked a lot like these slow turnover curves that I was just telling you about. And so this seemed really great. It seemed like mangroves were taking young carbon, they were transporting it to depth where it was being stored for a long period of time, meaning mangroves would be very effective carbon sinks. And that confirms kind of a lot of what is said in the blue carbon literature. However, it's never quite that simple. This is where our ramped pyrolysis and oxidation method comes in. This is referred to as the dirt burner in my lab, if anyone has heard of it. And so I want to provide you with kind of an ideal situation before I present you with our data. So this is what we end up expecting. So when you're looking at these plots, this will always be the amount of carbon dioxide that is emitted during the reaction. Our temperature increases on this axis, and then our ages go from younger to older. And so this method is based on the thermochemical stability of sediments. And so this dark black line, you can see the evolution of our carbon dioxide throughout the reaction. And then these boxes here are individual radiocarbon dates. So you can think of them as subsamples or subdates within a single sample itself. So what we really expect is that at low temperatures, we're going to have labile material that's going to be reacting first. And this labile material is also going to correspond to a young age. At higher temperatures, we're going to have the more refractory material that's burning off at a higher temperature, and that's going to correspond to an older age as well. So you end up with this nice spectrum of ages from young to old, from a low temperature to a high temperature. So we have our nice ideal example, and then we have our data. So I want you to focus again on these red boxes, and then these are our dates over here. So our boxes, our ages, all ended up being the same height, which means they're all the same age. And not only that, but they were all young. So they had a fraction modern greater than 1, which means they were all produced after 1950. And so this left us a little confused at first. It was definitely far from the ideal situation, but we realized in order to have this type of homogenization of our age spectrum, we had to have a rapid turnover time, which was contrary to what we had thought before. And so this must mean that carbon is entering and leaving our system so quickly that it prevents the incorporation of older material. So we would expect to see our older material kind of peaking over here, and we don't see that at all. And so this rapid turnover time in turn would mean that mangroves are really not the effective carbon sinks as we had previously thought. And at this point, if we really wanted to challenge this widely held paradigm, I figured it'd be good if we ensured that our methods that we are using are really great. So I wanted to look at our methods and other methods in the field to make sure we are really using the right things. So one method that is commonly used is compound-specific radiocarbon analysis. And so in a nutshell, compound-specific analysis, you have your sediment sample, you extract a series of compounds that can be anything from lignins to fatty acids, and in a much simplified manner, you get a radiocarbon age. And for me, I thought clearly there's no way these tiny compounds could be representative of this entire sample. Of course, ramp pyrox must be better. And so I decided to do my own experiment on our own method. So we take our sediment, we filter it down to less than 63 microns. That is what's run through our dirt burner, and we ultimately get a radiocarbon age. So after doing all of that processing, I had to ask myself, is this filtered sediment actually representative of the sample of the data we're getting? And I found out that really we're only dating 1% to 6% of our original sample. So I can't say definitively that this process is doing much better than what others in the field are doing. And so to wrap up with some implications. So mangroves, at least at our study site, we know are taking in some young carbon out of the atmosphere. However, we have evidence that this may not represent a long-term sink of carbon. And if this is the case, then the effectiveness of this carbon sink may be severely overestimated. However, I think we do need to expand the scope of the study to include sites that are across the globe to see if this is a local phenomenon or a global one. In addition, I think the radiocarbon and blue carbon communities need to decide on the best methods for answering this question. And so I think you hear the garbage in, garbage out a lot when it comes to modeling. But I think in this case, this applies here too, where we need to determine if we're using the wrong methodologies, will we be coming to correct conclusions, and test additional methodologies to ensure that we're accurately representing the samples we have. So with that, I'd like to thank these people and take any questions.